Today we're doing lipids, chapter 10. Key topics include biological roles for lipids, structure and properties of storage lipids, and structure and properties of membrane lipids, and signaling lipids. So, do you guys know what lipids are?
Fat.
Yeah. Basically. So fats. For the most part. So we have all different types. It's very diverse. And we have, you know what this is?
Hydrocarbon chain, carboxyl group.
Which is a...
saturated lipid.
That's unsaturated.
Well, this one's got a saturated hydrocarbon chain, but it's not saturated because it's got the carboxylic acid group on it, right? But this is a free fatty acid. You've heard of fatty acids? That's what that is. It's a fatty acid because of the long hydrocarbon chain and the carboxylic acid group. They come together and make triacylglycerols, which is, do you know what triacylglycerols are used for in your body? Any ideas? Huh? Not membranes per se. Diacylglycerol with a phospho.
Is it used for water retention?
Energy storage and insulation, right? So there's a ton of energy in all those bonds because they're completely reduced for the saturated and mostly reduced for the unsaturated. You see, this is unsaturated. You got a double bond there and a double bond there. You guys understood that, the definition of saturated versus unsaturated fatty acids? Anyways, I'm getting ahead of myself. This is just to show you the structural diversity in lipids. So we have free fatty acids, triacylglycerol, phospholipids. What's ubiquinone? You guys ever heard of that?
Coenzyme Q, but I don't really know what it does.
Yeah, coenzyme Q. So that's totally important for the electron transport chain. It shuttles electrons from the different subunits in the electron transport chain. Cholesterol, what's cholesterol for?
Steroids and...
Yeah, it's a precursor for steroids.
Used for signaling, used for...
And bile acids and membrane fluidity and whatnot too. It alters the membrane fluidity, right? So, okay, and then basically all lipids are basically organic molecules and they're characterized by their low solubility in water, which means they're relatively hydrophobic, right? So, functions include storage of energy, and we just saw that they're highly reduced compounds of a hydrophobic nature, so you can store a massive amount of energy in a small space, and what's that going to do? Like, why is it good that it's separated from the water? Any ideas?
The water heats up when you use the energy. And...
Not that the water heats up, but you know, you ever heard osmolarity?
Yeah.
So when you have all this energy that's away from the water, it's not going to impact that osmolarity of storing that energy. It's kind of separated from all the solutions where you have chemical reactions and life taking place, right? It's just separated. So hydrophobic nature, and then it packs well together so you can store a lot of energy in a small volume too. Okay? And you don't contribute to any osmolarity imbalances. Insulation from the environment, it's low thermal conductivity, high heat capacity, meaning it'll absorb a lot of heat, and mechanical protection, so it can protect you from shock. That's why those football players are always kind of chubby, right? Water repellent, it's hydrophobic, so there's certain lipids that, you know, keep water out of things, like waxes, right? So, and then buoyancy. So mammals, marine mammals, often have a lot of fat because it makes them buoyant. You know, lipids are less dense than water as far as weight to volume, so lipids will float on the water. So if you have a lot of fat, that'll make you more buoyant. So membrane structures is another function. So all our plasma membranes are composed of lipids, basically. What kind of lipids?
Bilayer.
Yeah, but what makes up a bilayer? They're not triacylglycerol. They're phospholipids, right? Because they have a charged head group that's going to interact with the water, and then the acyl tails. Cofactors for enzymes, so vitamin K and coenzyme Q are both cofactors. Actually, coenzyme Q shuttles electrons back and forth from the different...
Is there a difference between the mechanism of the lipid with its unsaturated and saturated? Because I know that coenzyme Q is very saturated and it's got...
Well, coenzyme Q, it's a lipid-soluble electron shuttle. So it shuttles within the membrane electrons from one of the
polar ends to...
Not necessarily a polar end. It shuttles it from one complex like 1 to 3 and complex 2 to 3. Or not 2 to 3, but 3 to 4. And then, I forget. I didn't study up on my electron transport chain today. But it's not so that's so saturated. It is pretty saturated, but you have branch, and then you have a place up here. You can add hydrogens and take away hydrogens depending on, it'll accept the electrons or take the electrons. So it's all pretty much soluble in the membrane, that one is. I'm going the wrong way. Pigments, oftentimes pigments or lipids, so the color tomatoes, carrots, pumpkins, and birds. And then antioxidants also, vitamin E is an example of an antioxidant. That's a lipid-soluble antioxidant. And this shows you the pigments. And just the little differences causes a difference in the wavelength of light it's going to absorb, which will alter the color it reflects back, right? So basically, lipids they're classified based on their structure and function. So we have storage lipids, which are basically neutral. They're triacylglycerols. You have one glycerol with 3 fatty acids, and they form. Do you know what kind of linkage that would be? Anybody know the structure of glycerol?
Triglycerides.
Not triglycerides, just glycerol. So glycerol, it's an alcohol, right? You have 3 carbons with 1 alcohol on each one. Which makes it fairly soluble in water like this, right? But triacylglycerol, you have 3 fatty acids, and what kind of linkage to an alcohol would an acid be? No, not glycosidic. That's sugars, right? Remember organic chemistry?
The bond that holds the
Carbonyl.
Not a carbonyl.
hydroxyl? No, wait, they're talking about...
The kind of bond, like if we have
An ester?
yes, an ester.
Oh.
So basically, it's a dehydration. You remove water and you have an ester bond. So ester bonds, what you get in triacylglycerols. Same thing with phospholipids. You have ester bonds with fatty acids. And then we have sphingolipids, where instead of glycerol forming the backbone of the structure, we have sphingosine forms the backbone. And again, sphingolipids, glycolipids, what do you think glycolipids have in them?
Sugar.
Right, sugars. And then there's archaebacterial and ether lipids. So if it's an ether lipid, what kind of bond is that? Instead of fatty acids, you have what?
Ether linkage.
Yeah, what would make an ether linkage? Yeah, there's an oxygen there, but not a double bonded. The ether linkage is just going to be basically if you link 2 alcohols and remove the water, that would make ether, right? So it's just an oxygen. Yeah. So, okay. So fatty acids, your most basic, they're carboxylic acids with hydrocarbon chains containing between 4 and 36 carbons. Almost all natural fatty acids have an even number of carbons, and natural fatty acids are unbranched for the most part. Saturated, we already discovered, or you guys already know, right? Just means that there are no double bonds between the carbons in the chain. So they're completely reduced, they're saturated with hydrogen, right? Monounsaturated means you have one double bond. Polyunsaturated means you have more than one double bond, right? So, omega-3 fatty acids, have you guys heard of those? Yeah, so we need them. And basically, this one is an example of an omega-3. You count up from the last carbon. So it'll be 1, 2, 3, and you have the double bond there. But that's not normally how you do, like, fatty acid nomenclature. For example, this is like 18,1 cis-9 octadecanoic acid. So the 18 and the 1 means it's 18 carbons long, and then there's one double bond, and they put a delta 9 because that double bond is between the 9th and the 10th carbon. So pretty simple nomenclature. And then for this one, 25, because you have 5 double bonds, 1, 2, 3, 4, 5. 5, 8, 11, 14, 17 for the double bonds. And then it's, I don't know how to say this, eicosapentaenoic acid. Is that right?
That's right. How did you say the counting goes from the end?
The which one?
From the opposite end of your...
Yeah, so for like the omega-3s or omega-6s, rather than starting here at the carboxylic acid, they start at the end for the omega designation. Like you've heard of omega-3, it's 3 carbons up from the last. Yeah. And then omega-6 fatty acids have it right here. So they call this the omega-3. It'd be an omega-3 because there's 3 carbons from the end. Omega-6s would be, this would be a double bond here, it would be here instead.
It would be named the other way too, that's why they have the numbers.
Yeah, this is the more official way to name fatty acids, but then, you know, you've heard of omega-3 and omega-6s, that's where that gets that designation, is counting up from the omega carbons. Okay? So this just shows you some more of the fatty acid nomenclature. For 12 carbon skeleton, it's in dodecanoic acid, or lauric acid is its common name. Most of the time you'll see these as their common names. Palmitidic acid is probably the most common, it's 16 carbons, and saturated, and so forth. And then you get the unsaturated palmitoleic acid, and you have, looks like delta 9, I can't read that, but 16 and 1 double bond, so it's delta 9. So solubility decreases with the chain length as the chain length increases. And then, why is that? Huh? Well, yeah, van der Waals interactions is going to probably have more to do with this, right? The melting point. But with the solubility, increased chain length, there's more carbon out there that's going to have to interact with the water. So you're going to disrupt well, you'll have to make ordered water, right? So around that. So as that gets longer, there's more ordered water around that. So melting point, basically it's going to decrease as the chain length decreases, and that's because of the interactions with the van der Waals forces, right? Shorter van der Waals forces means that they're going to, shorter chains means the van der Waals forces will be lower, so you'll need less energy to split them apart, right? And then decreases, the number of double bonds increases. And why is that? Any ideas? One thing I think I forgot to mention is that pretty much all unsaturated fatty acids in nature, or not all of them, but pretty much, are cis double bonds. Do you remember cis and trans?
So all fatty acids are cis?
For the most part, yeah. Not all of them, because we can make trans fats. You've heard of that?
Yeah.
So That's where that comes from, the trans fats. But the majority, pretty much all natural unsaturated fatty acids are going to be cis. So as you increase the number of double bonds, it also, decreases the melting point. Anybody have any ideas why that is?
Because it's more
Easier to break apart.
Well, it's more rigid, but also it's cis, so it has a kink, right? Whereas these long straight chains, they're less rigid, but they can pack really, really close together, and they'll complement each other quite nicely. Whereas if you have some of these stuck in there, they can't pack as closely, right? Because it's rigid, and that this double bond is causing it to have a bend. And you end up, if you have a mixture of saturated and unsaturated fats, they can't pack as close because of that kink. Whereas pure saturated fats, you'll get really nice packing, so it'll have a higher melting point too. Because you get more, when you have packing like this, you have more van der Waals interactions between the neighboring molecules, right? So that's why you have more saturated fat in animal foods than you do when you get plant foods, right? Do you understand why that is? So plants, what temperature do they normally grow at?
30 degrees Celsius.
Huh? Yeah, like room temperature, whatever temperature the environment is. So if the plant's growing out like in 20 degrees, you can alter the membrane fluidity by adjusting the amount of double bonds there. So plants grow in a cooler environment, so to have a more fluid membrane in that environment, they're going to have a lot more, unsaturated fatty acids. Whereas animals, or like for mammals anyways, we're growing at 37 degrees, so we're much warmer than a lot of the plants would be. So we'll have more saturated fat in our tissues because that'll, it regulates the membrane fluidity, basically, how many double bonds you have in those lipids. Does that make sense, kind of? So that's why most animal fats at room temperature are solids, like, lard. It's a solid, and whereas most of the oils you get from plants are going to be liquid at room temperatures. So trans fats, we just mentioned these. They're formed by partial dehydrogenation of unsaturated fatty acids. So if you just do the partial, you're breaking those cis bonds for the most part. You partially hydrogenate. So a lot of those fatty acids have multiple unsaturation points, but you partially dehydrogenate, you're basically going to break all the double bonds while you're doing this process, and it'll allow it to form a trans when it forms back up. Does that make sense? It'll form in a trans conformation because it's thermodynamically that's less steric interference in a trans conformation. So it's done, they do this to increase the shelf life or stability at higher temperatures than, so if you're going to use oils, margarine will keep longer, or trans fats keep longer than regular, like olive oil or your liquid oils, right? Those oils are subject to oxidation, and one of the reasons they're subject to oxidation is all those unsaturated points are places for oxidation to occur. But they're also liquid, so that liquid allows for reactions to occur too. So trans fatty acids can pack more regularly and show higher melting points than at cis form. Consuming trans fats increases the risk of cardiovascular disease, and we've all known that, right, for a while. So current trend is to reduce the trans fats in foods. So. And this is just a table showing you the different, amounts of trans fats that we see in different foods. So triacylglycerols are basically non-polar. They're all triple ester linkage with glycerol, right? Solid ones are called fats. The liquid ones are called oils. And the primary storage form of lipids in the body is these body fats. Less soluble in water than fatty acids due to the lack of a charged carboxylic acid group. And they're less dense in water, which means they'll float. And that's where you get the buoyancy in marine mammals, right? What do free fatty acids form in solution?
Micelles?
Yeah, micelles, right? So, and this is the overall structure of a triacylglycerol. If you notice, there's 3 carbons. So here you can have 3 different fatty acids bound to the glycerol in ester linkages. And this would be known as a mixed triacylglycerol if you have different fatty acids listed. So fats provide efficient fuel storage. Fatty acids carry more energy per carbon because they're more reduced. Especially saturated fatty acids are the most reduced. So you'll get more energy from saturated fatty acids per gram than you will with unsaturated, right? Fatty acids carry less water per gram because they're nonpolar. So they don't have that weight of water added along. Like if you had starch or glycogen, it's going to carry water molecules with all those sugar molecules. It's actually one big sugar molecule, right? The polysaccharide. But water will be there because of all the polar, alcoholic groups. Glucose and glycogen are for short-term energy needs, and they can deliver those energy quickly, whereas fats are longer-term. We acquire fat over months to years, and it's if we go starving, that's when we start to burn the fats that we store. And this is just the adipose tissue. So it shows you what white adipose tissue, you have these big fat globules within the cells. So waxes are interesting. They're esters between long-chain saturated fatty acids and saturated and unsaturated fatty acids with long-chain alcohols. So rather than, like a glycerol has 3 alcoholic groups, waxes, you have just 1 fatty acid and 1 long-chain alcohol. And that's where you get the wax. So they still have a head group. A polar head group is going to be that ester bond, right? And they're very hydrophobic, which is what makes them so good at keeping water out of things, which is what waxes are used for in nature. So they're good for storage of metabolic fuel and plankton, their protection and pliability for hair and skin invertebrates, and waterproofing of feathered birds, protection from evaporation in tropical plants and ivy. And it's used for, people for lotions and ointments and polishes, basically.
Is it just that esters are the difference in fatty acids and the waxes, and that's the only difference that we have?
In the what?
In the waxes.
The only difference between waxes and triacylglycerols is you have 1 fatty acid and it's in an ester with a fatty alcohol.
Okay.
Rather than a glycerol, which has 3 alcohol groups on it.
Okay.
So beeswax is the same kind of structure. So structural lipids and membranes, they're polar head groups, right? And non-polar tails attached to usually fatty acids. Diversification can come from modifying different backbones. And I showed you at the beginning, you have, the most common backbone is glycerol, right? And the other one, do you remember? Probably not. But sphingosine or something like that. So, and you change the fatty acids, so you can have multiple arrays of different fatty acids, modifying the head groups also. There's a number of different head groups. Anybody ever heard of phosphatidylcholine, phosphatidylserine? Those are all different head groups on the fatty acid. Phosphatidyl and anidosine, I don't know if I say these right. Properties of the head groups determine the surface properties of the membranes. Different organisms have different membrane lipid head groups compositions, and different tissues have different membrane lipid head group compositions. So there's primary constituents of all membranes are glycerophospholipids, which means you have glycerol phosphate or L-glycerol 3-phosphate that gets that L designation. The phosphate group always, because normally if you just have glycerol, it's basically the same front and back, right? There's no, but once you add the phosphate, it gives this number 2 carbon chirality, right? So the phosphate always is given the number 3 carbon designation. So it's L-glycerol 3-phosphate or SN-glycerol 3-phosphate. So you can say glycerol is prochiral. Have you ever heard that term? So before it's modified with the phosphate, you can say it's prochiral. So the head group's always charged at physiological pH, right? Phosphate has 3 Kas, right? 3 pKas. Huh?
This phosphate has 3 pKa.
Phosphate has 3 pKas, phosphoric acid, which
the hydrogen.
because of these oxygens. So if you didn't have this in a phosphoester linkage, this would be protonated. Right? I think the pKa is around 12.4 for that last one. So general structure of glycerophospholipids, unsaturated fatty acids are found normally connected to the number 2 carbon, and then the highly polar phosphate group may further esterify with an alcohol, such as the substituent groups are called the head groups. So you get another ester with a different head group, and that's where you get phosphatidylcholine, phosphatidylserine. So phosphatidylserine contains a serine esterified there, right? So you have the fatty acids on the number 2 and 3 carbons, and then a phosphate with a head group attached to the phosphate.
So it's just the glycerophospholipids and cell membranes that get the number 3 notation is what you're saying, and the unsaturated ones get the number 2.
Well, yeah, the glycerophosphate, it's always on the number 3, the phosphate. And then the unsaturated fatty acids typically go to the number 2 carbon. Oh, did I go the wrong way again? Right. Like this, we have linoleic acid, and it's attached to the, has an ester with the number 2 alcohol. And then saturated are often on the number 1s. These are some of the examples of glycerophospholipids. Phosphatidic acid is just the phosphate as a head group. Phosphadylethanolamine, you have an ethanolamine group. And that's what you have. You have the ester with the phosphate and the OH on the ethanol. And then ethanolamine. This amine group gives that head group a positive charge, which will make the overall lipid neutrally charged, right? Do you see how that works? Because the phosphate, if it didn't have the ester, it would have 2 negative charges. You have the ester with the ethanolamine, so that gives it a -1 charge because you still have 1 negative charge on the phosphate, but then this amine group is positively charged on the ethanolamine, which that counteracts so overall net charge of that lipid would be 0. Phosphatidylcholine, it's very similar to ethanolamine, but choline has a methyl group on each one of the amines. So that also has a 0 charge, because they don't show you that this tertiary nitrogen is going to have a positive charge, right? So phosphatidylserine, you have an ester group with a serine molecule, and that's a -1 charge. Phosphatidylglycerol, you just have a glycerol there as another head group. Phosphatidylonidazole 4,5-bisphosphate. So anidazole is like a sugar alcohol, and you have 2 phosphates added to that. And this is an important signaling molecule we'll learn later. Cardiolipin is a phosphatidylglycerol head group. So, and does anybody notice something else for this cardiolipin? Oh, I'm jumping ahead. Sorry. So phosphatidylcholine is the major component of eukaryotic cell membranes. Many prokaryotes, including E. coli, cannot synthesize this lipid. Their membranes do not contain any phosphatidylcholine. See, here you can see the positive charge. I don't know why they didn't put it on the last table. But any tertiary nitrogen is going to have a positive charge, right? Ether lipids can occur. So what's an ether bond between? Yeah, so two alcohols basically, right? So so which do you think are going to be more stable? Ethers will be. Yeah. Because with an ester you have this carbonyl group still there out in the wind, so it's a great nucleophile, right? Or is that no electrophile?
Yeah.
I just remember one.
You pointed that out on the last slide. It's got the 2 carbonyls in there.
Yeah.
So right next to the R groups, you've got two carbonyls that are just floating at the end.
Yeah. So these are both ester linkages, so you can get an attack here of water, right, which will substitute, you know, for the alcohol here, and it's easy to cleave esters, whereas ethers aren't as electrophilic. So, and that's why they're resistant to cleavage by common lipases, but there are some specific lipases that will cleave ester linkages. They increase membrane rigidity, and they're sources of signaling lipids, and they may be antioxidants also. So one example of a plasmalogen is this. Typically, you have an ether-linked alkene, so it's an alkene right next to the carbon that has the alcohol, or it includes the carbon that has the alcohol. And then you have an ethanolamine head group, and then another fatty acid, and that's what makes up plasmalogen. One of the more common ones that we see in human physiology is this. It's platelet activating factor. So this plays a role in inflammation and clotting. If you've, what's that? Toxic shock syndrome, this is involved in that. Have you guys heard of that before? So aliphatic ether, here you have an ether, and this time it's the ether-linked alkane, so it's saturated, and then an acetyl ester group, so you have an acetic acid rather than a long-chain fatty acid for the ester on the number 2 carbon, and then choline is a head group. So sphingolipids are a different type of phospholipid, so rather than having glycerol as the backbone, we have sphingosine. It's a long-chain amino alcohol, sphingosine. And a fatty acid is joined to the sphingosine via an amide linkage rather than an ester linkage, usually seen in other lipids. Polar head group is connected to sphingosine by a glycosidic or phosphodiester linkage. And the sugar-containing glycosphingolipids are found largely on the outer face of plasma membranes. So these sugar-containing glycosphingolipids, they're on the outer face of the plasma membranes. So you have these plasma membranes with a bunch of sugars attached to it. So what do you think that'll be involved in?
Transportation and signal transduction.
Well, so a lot of these are involved in immunogenicity, like for tissue. So like your blood types depend on the sugars that you have attached to a sphingosine. So this is your basic structure of sphingolipid. This here in pink, or is that's pinkish or peach? Huh?
Salmon.
So that's the sphingosine. Yeah, salmon is what you said, right?
Pink. Go with it.
Pink or salmon. So this is sphingosine. You see there's an amide linkage with a long chain fatty acid, and then you're going to have a different head group, and the different head groups make what it is. If you just have a hydrogen instead of a head group there, it's called ceramide, which is the basis to make all the other sphingolipids. One includes sphingomyelin, where you'd have a phosphocholine head group. Where do you think you find lots of sphingomyelin?
Muscle.
And bones, teeth.
Where do you find myelin in the body?
I thought that was.
Muscle?
No.
Oh, wait, no, sorry, bone. That was the trunk. Oh, yeah.
Nerves. Myelin sheath. You've heard of that? So you find a lot of sphingomyelin in those myelin sheets. Neutral glycolipids like glucocerocerebroside. So this is cerebroside. You only have glucose in the glucocerocerebroside. So once you attach a sugar for the head group, it becomes a cerebroside or a globoside, where you have multiple sugars. Ganglioside, GM2 is a specific ganglioside, and you have complex oligosaccharides. There's multiple sugar groups in the gangliosides. And they don't go into the specifics. So, but ceramide, like I just said, is basically a sphingosine and an amide length fatty acid, the sphingosine, without a head group. That's ceramide. You put phosphocholine on it, and you have sphingomyelin, which is, like I had just said, found in the myelin sheath in the nerve cells. So they're structurally similar. If you look at sphingomyelin and phosphatidylcholine, they're really similar looking, right? So you still have the phosphocholine head group. The only difference is you have this amide group in the sphingomyelin versus just another unsaturated free fatty acid with the phosphatidylcholine.
Doesn't determine its unsaturation or saturation, because that's the amine, it's saturated, that's the...
I think you could have unsaturated, but for the most part I've only seen saturated in here. And then you have an the sphingosine itself is unsaturated there. If you look at the sphingosine molecule.
Yeah, because in the book at least I couldn't find one that's sphingo with the nitrogens. Is that because of the charge on the nitrogen, or is it specific to anything else?
Nah, just probably specific to the enzymes that catalyze the amide linkage formations. So like I was saying, blood groups are determined in part by the type of sugars located on these head groups. So depending on what enzymes you have coded in your DNA will make up your blood group based on, because that's the different sugars that's going to add. So individuals with no active glycotransferase will have the O antigen. So you don't have the enzyme to add the other sugars to that sphingosine. Individuals with a glycosyltransferase that transfers N-acetylgalactosamine, that makes them A blood group. And then if they have a glycotransferase that transfers a galactose group, that gives you the B blood group. So your antigens look like this. You have the sphingosine molecule with the fatty acid. B antigen has, what was that? Galactose group. So that round is a galactose group. A antigen has the N-acetylgalactosamine. So that means you have the amine group. It's a modified galactose. And then O doesn't have that at all. So defects in the turnover of lipids lead to a number of diseases. Anybody ever heard of Tay-Sachs disease? No?
Yeah, well, I've heard of it before. I don't know the specifics.
Yeah, so that's if you don't have hexosaminidase 1, you can't. You'll lack like GM3, the ability to make GM3 from GM2. So it limits your turnover of your gangliosides. So sterols and cholesterol. So steroid nucleus has this forward fused rings. You guys are familiar with cholesterol? And this serves as the basic structure to produce all your steroid hormones. And what else? Do you know?
Steroids?
What else is cholesterol used to make that your body uses that's not a steroid hormone? No. How about, you ever heard of bile salts? So so for the absorption of fat. So the steroid nucleus is almost planar in this configuration. It's almost planar. So the cholesterol and related sterols present in the membranes of most eukaryotic cells modulate the fluidity and the permeability of the membrane. They'll thicken the plasma membrane, and most bacteria lack sterols altogether. Mammals obtain cholesterol from both food, and we can synthesize it ourselves in our liver. Did anybody know the enzyme, the rate limiting step? What blocks that rate limiting step? What that synthesizes cholesterol? Come on, you guys both work in the pharmacy.
I know.
CYP4. Those are the oxidases, right? Mixed oxidase system, which is involved in making the convert. There's a mixed system oxidase, I think, adds that OH groups and cholesterol formation, but it's not the one we're talking about. HMG-CoA reductase.
Yeah, we use that one all the time, man.
Yeah.
But what drugs block that, you know? Statins?
Oh, okay.
Yeah.
Yeah.
What was that?
So, cholesterol, we find it bound to proteins, and it's transported to tissues via blood vessels, right? So how's it transported around the cell, do you know? Lipoproteins, are you familiar with those? So you have your HDLs, LDLs, VLDLs, and chylomicrons, and IDLs too, right? Huh?
They're just like little micelles that...
Yeah, basically they're little micelles that have like a protein that actually has alpha helical sections with a proline that causes a little kink in the alpha helix, and it kind of forms a alpha helix that goes around a fat globule that contains cholesterol esters and triglycerides, right? So, and many hormones are steroid derivatives also, we know that, right? Steroids are oxidized derivatives of sterols. Steroids have the sterol nucleus but lack the alkyl chain found in cholesterol. They're more polar than cholesterol, right? Because they're lacking that alkyl chain. Steroid hormones are synthesized from cholesterol in the gonads and adrenal glands, and they're carried through the body in the bloodstream, usually attached to carrier proteins. Many of the steroid hormones are male and female sex hormones, right? And this is basically a figure showing you the structure of all the steroid hormones. Testosterone, beta estradiol. You can see they lack the alkyl group. You have cortisol, that's a stress hormone. Aldosterone, brassinidyl. I'm not familiar with that one, are you? Prednisone or prednisolone and prednisone, right? Biologically active lipids are present in much smaller amounts than the storage or structural lipids, and they play vital roles as signaling molecules between nearby cells. Lipid-soluble vitamins also A, D, E, and K. So, arachidonic acid is basically the basis for the synthesis of a number of signaling lipids, and you guys have heard of prostaglandins. So they're involved in inflammation and pain. So inflammation and fever, thromboxans, formation of blood clots, and leukotrienes, smooth muscle contraction in lungs. So basically you have arachidonic acid is the precursor for leukotriene. It's also the precursor for thromboxan A2, and then also for prostaglandin E1. And this is where your NSAIDs act on, right? So your aspirin, ibuprofen, acetaminophen all act on the synthesis of prostaglandins and thromboxans from arachidonic acid. You see, this is just a free polyunsaturated fatty acid that is the precursor for all of these.
So acetaminophen's not an NSAID, so does it work?
It's just the anti-fever reducer thing.
It works.
It works the same way as
Yeah. So aspirin, do you know the structure of aspirin?
It's like salicylic acid and something else on it, right?
Yeah, it's acetyl salicylic acid, or acetyl salicylate. So basically that works by adding just acetyl groups onto the enzymes that do this, which then blocks that active site from performing its function. So, and then the other NSAIDs do the same, but they don't add some of them, I think.
Yeah, doesn't ibuprofen work by like a second passage theory or something like that?
A second passage?
Like, I don't know, I can't elaborate, I guess. But like it has to be metabolized and then it's something else again or something.
Oh, like it's a pro-drug before it works?
I don't remember that.
I don't know.
Metabolite, yeah, that's what it is. Like the metabolite.
The metabolite is the active form.
Yeah.
So it's a pro-drug.
Yeah, I guess.
It must be. I mean, yeah, if it's not the active. A lot of drugs are delivered like that because you know.
Well, when I'm thinking of a pro-drug, I'm thinking of like Vyvanse, and I don't think it's classified as like a pro-drug. I'd have to read. I'm not.
Probably not classified, but if the active form is what it has to be metabolized before, it's kind of is a pro-drug, just like Vyvanse is a pro-drug too. Vyvanse has what it's basically has, I forget what amino acid, and an amide or ester bond. I forget. But vitamin D regulates calcium uptake, right? So, and we all know we can make it with UV. So 7-dehydroxycholesterol gets converted after UV exposure to cholecalciferol or vitamin D3, which will then go on to become alpha-1 25-dihydroxy vitamin D3 or calcitrol. Vitamin A or retinol, where do we use that?
Vision?
Yeah, retinaldehyde or retinal. So basically it gets cleaved from beta carotene, you have vitamin A, retinol, get oxidation to the alcohol, and you have 11-cis retinal. If you see, you have a cis bond here. When it absorbs visible light, it'll excite these electrons, allow it to rotate around that bond, and then when it drops back down to its ground state, it forms all trans retinal then, which will then cause a conformational change in a protein, which causes a G protein signaling that ultimately tells your brain that you absorbed a photon of light in that one cell. Basically how your vision is created, right? And this will go on further, oxidation to form retinoic acid, and then that's used in signaling. You can actually take stem cells, if you treat them with retinoic acid, you can convert them to neurons. So vitamin E and K and other lipids are antioxidants. So vitamin E is so it's lipid-soluble antioxidant. It has this aromatic group that can absorb electrons. Vitamin K, it's used for blood clotting. They give you that shot first thing when you're born to make sure you're going to be able to clot your blood. Warfarin is an anticoagulant. It looks similar to vitamin K, kind of blocks vitamin K's function. Ubiquinone, or the coenzyme Q, that's used in your electron transport chain. So it shuttles electrons between different groups in the electron transport chain within the membrane. Plastoquinone, it's basically the plant version of this, but rather than the electron transport chain, it's used in photosynthesis to shuttle the electrons down the Z scheme, I think it's called. And dolicol is a sugar carrier that's lipid soluble. So polyketides are biologically active lipids, and they're used in medicine. So we have erythromycin, so we have sugar molecules, and then a polyketide basically structure. Amphotericin B is an antifungal, and then lovastatin. It's a statin drug, so this is actually used for what?
Cholesterol.
Yeah, HMG-CoA reductase inhibitor.